Digital Champions, long time since we have met. How are you all? I hope every one of you are doing really good. I also know one more thing, that is your exams are nearing and you might be worried about what to study, where to study from, and lots of questions might be there. I'm just here to answer at least one of your questions, and that is how to deal with this chapter, which is Microbes in Human Welfare, a chapter from biology. So I hope I could help you really well in this. Now listen to me, this chapter is really fun, really interesting, and you don't have to study all of it. Okay, so for example, you're getting stuck at one place which is not very much useful for giving you marks. You can just move on to the next thing. That is what I meant to say. So I'm going to tell you what is more important and what is not. Okay, so you can skip accordingly, you can study accordingly, which is more important rather than skipping, right? So chalo. So the first part of the chapter, just have a quick read of it. Okay, don't invest too much of your time into would say having a read of it. When you come to this particular page, that is page number itself is not getting loaded. So let's wait for it to get loaded. Just give me a minute's time, and I'm so sorry if my voice is cracking or something like that. My throat is down. Okay, so we have the first bacteria, that is lactic acid bacteria. You should be knowing about its use. What is the use? Basically, it converts milk into curd. Also, it does one more thing. It adds nutritional quality. What nutritional quality? Basically, it increases vitamin B12 content in the curd also. So, vitamin B12 content is increased. Apart from that, it is the one that converts what? Milk to curd. Okay, so LAB, that is lactic acid bacteria, produce acids. What does it produce? Lactic acid bacteria. So it is going to produce certain acid, and this acid is going to coagulate and partially digest the milk protein, that is casein. So in the milk there is a protein which is known as casein. This casein which is there, it is partially digested, which is why there is its shape and everything get disoriented, which is why we get curd. Okay, we get small lumps which is known as curd, my dear children. So in this lactic acid group of bacteria, you have lactobacillus as an example as well. So you have to be very sure about all these names. It's very important. Now talking about the baker's yeast, or what do you call as Saccharomyces cerevisiae. Okay, which is just the yeast. It is also known as baker's yeast because it is used in making of bread, my dear children. So you should be knowing what is that something which is used in making bread. It is Saccharomyces cerevisiae, as you can see over here. Okay, cool. Now apart from that, have you heard about Swiss cheese, my dear children? So in Tom and Jerry, Jerry used to always sneak out and eat the cheese, and this cheese has big holes. Well, why there are big holes, my dear children? This is a question. Why there are big holes in Swiss cheese? Who is responsible for that? So there's a bacteria whose name is Propionibacterium shermani. Okay, Propionibacterium shermani. In Hindi, Sharmana means you are shy of something, right? So Propionibacterium shermani, basically it releases large amount of CO2, which is why there are holes in the Swiss cheese. And apart from that, there is example of when I don't want it to zoom, it will be zooming. When I want it to zoom, it does not zoom. Yeah. Then there's an example of Roquefort cheese. Now here, the Roquefort cheese is ripened. Okay, it is ripened because of a fungus. This is again a question that is asked. Who is responsible for the ripening of Roquefort cheese? It is what fungi? Now they have not mentioned what fungus, this fungus, or that fungus. You just have to know it's a fungi. Okay, so about yeast we spoke. Yeast is a fungus, my dear children. Apart from that, we spoke about Roquefort cheese. It is by another type of fungus. It's not yeast, it is some another type of fungus which is responsible for its ripening. For Swiss cheese, we spoke about the large amount of CO2 being released, which is why there are holes, and this is by a bacteria, that is Propionibacterium shermani. Okay, and lactic acid bacteria, I told you, it increases the vitamin B12 content, and also it converts milk into curd. Now, going ahead, what do we have? We have the fermented beverages. Okay, we have the fermented beverages. So over here, Saccharomyces cerevisiae, again, it is also used for bread making and commonly called as brewer's yeast. It is also known as brewer's yeast. So over there, we saw it as baker's yeast. Now it is known as brewer's yeast. Why? Because it is used in fermenting malted cereals and fruit juices to produce ethanol. Ethanol is an alcohol. Okay, so what do? Basically, this malted cereals as well as fruit juices, they are fermented to produce ethanol with the help of the same yeast which is known as Saccharomyces cerevisiae, but over here you call it brewer's yeast because it is dealing with brewery, that is making up of alcohol, right? So it helps in fermentation of malted cereals and fruit juices. Again, a straight question. Okay, what is that helps in the fermentation of malted cereals and fruit juices, and what does it produce? Basically, it produces ethanol, and who is responsible? Saccharomyces cerevisiae. Okay, now apart from that, the question is asked, what type of beverages are produced without distillation and what type of beverages are produced with distillation. So wine and beer are produced without distillation, my dear children, whereas whiskey, brandy and rum are produced by distillation of fermented broth. So distillation basically means they are going to vaporize it, and whatever alcohol content is there, that will be remaining, that will be more, that might have more amount of alcohol. So the one that will undergo distillation, it will be having too much amount of alcohol content, whereas the one that does not undergo distillation, it may have less amount of alcohol content. So which is why wine and beer, we know it has less amount of alcohol content, whereas whiskey, brandy and rum, all of them, they are produced by distillation of what? Fermented broth. So obviously the alcoholic content would be too much, so concentration will be way too much. So this is a question, so use your brain and just answer. Wine is something that everyone has, okay, because probably it does not have too much alcohol. Same goes with beer also, okay, it has alcohol but comparatively lesser than whiskey, brandy and rum. Okay, now this question, who was the person who was behind the production of penicillin? Okay, penicillin is an antibiotic, okay, and it was produced by Penicillium, it is produced by Penicillium notatum, which is a mold. That means you're talking about a fungus. So you should know that penicillin is an antibiotic, as in it is used to kill the bacteria, and it is produced by a fungi, which is Penicillium notatum. Okay, and who found it? Alexander Fleming was the person who found it. How? Because he was doing an experiment on Staphylococci bacteria. He was working on Staphylococci bacteria and he noticed that one of his unwashed culture, okay, one culture was not washed. In this point of time, still the Staphylococci could not grow over here. So he was like, why is it not able to grow over here? Then he found that, okay, there is another mold over here which is growing, which is Penicillium notatum's mold, and this might be probably releasing something, which is why the other guy is not able to grow. So that is how it was discovered. But later, okay, its actual role as an antibiotic and everything was discovered by another guy who was Ernstein and also Howard Florey. So Ernstein and Howard Florey came together. They gave rise to the antibiotic penicillin. They said that there is an antibiotic, that is penicillin, and it is very much important, and it could be helping in the treatment also. So basically these three guys, Alexander Fleming, Ernst Chain as well as Howard Florey, all of them got a Nobel Prize for the same. Okay, so just go through it, it's a fun thing, but you should be knowing about this. Okay, so always, what is the product that is being produced? For example, penicillin over here, and what is the organism who is producing it? So it is Penicillium notatum, and you should be knowing, is it a fungi, is it a bacteria, what all? It is a fungi. Okay, it is a fungi. Whenever you are talking about mold, my dear children, you are talking about fungus over there. Going ahead, what do we have? Okay, now from here, you would be asked, sorry, what is that something that produces citric acid, my dear children? So again, it's a fungus, and the name of the fungus is Aspergillus niger. Now, who is the person who is the organism that produces acetic acid? It is Acetobacterium aceti, my dear children, which is a bacteria. Now, what is that something that produces butyric acid? It is Clostridium butylicum, which is again a bacteria. Lactobacillus is the one that produces lactic acid. So again, for all the acids name, you have to remember who is producing it and which category does it belong to. So you can write it down, all of them, they are bacteria. Only one guy is fungus, which is Aspergillus niger, which is producing citric acid. Okay, Acetobacter aceti, name has something common with acetic acid. Clostridium butylicum, again with butyric acid, the name has something in common. Lactobacillus has a common thing with lactic acid, so you can remember it. Now, yeast Saccharomyces cerevisiae is used for commercial production of ethanol. I told you, for the production of alcohol, you need this particular yeast, that is Saccharomyces cerevisiae, which is also known as brewer's yeast. Now, for removing oily stains, now oil is what? Fat. Okay, oil is what? Fat, and fats comes under what? Lipids, my dear children. So can I remove lipids with the help of lipases? Yes, I can remove lipids with the help of lipases or lipases, my dear children. So please remember, this is a very important thing. So how is this removed? You should know it is lipases that is responsible for removing it. Now, from here again, so all this thing, I hope you will remember the name. If you are not able to remember the name, just solve so many questions, you will be able to remember. That is how I remembered. So while I was making the videos for Brahmastra, while I was doing some other other questions for your MCQ thing, and that's when I practice a lot of questions, and that's how even I remember it. Being a teacher, you won't trust me. You might think that I know everything. Teachers anyway will remember everything. No, my dear children, we also have to remember a lot of things, and we are also human beings, so we tend to forget a lot of things. We make so many mistakes as well. So that's why I am telling you, it's okay to make mistakes, but how can you remember in a better way? By solving lots of questions and also make proper notes and just go through it before you go to sleep. So you'll get an idea of this. Okay, now over here again, bacterium streptococcus. Okay, there is streptococcus bacterium that releases something that is known as streptokinase, and this streptokinase is acting as a clot buster. What do you mean by clot buster? It is responsible for busting the clot which is there, okay, for removing the clot which is there in the blood vessels. And these clots, okay, if they are there, it might lead to what? Myocardial infarction, leading to heart attack as well. So you might be asked like questions like this. I hope the question is here. The question is not here. I didn't take that. No, it's not here. Okay, so listen to me carefully, okay? So the question comes, what product acts as a clot buster for removing clots from the blood vessels of patients who have undergone myocardial infarction leading to heart attack? So you should be knowing it is streptokinase, which is the product, and it is being released by a bacterium, that is streptococcus. Cool? Coccus as in drought. Now the next product is cyclosporin A. Cyclosporin A, my dear children, it is produced by Trichoderma polysporum. This is again a fungi. Trichoderma polysporum is a fungi that produces cyclosporin A. What is the use of cyclosporin A? For example, you take kidney from me. In your body, this kidney is a foreign thing. Now, but you need the kidney anyways. Okay, you need the kidney anyways. So, but your body should be suppressed a bit, so that it does not fight against my kidney and just kill it off. So for that, I need to have immunosuppressive agents, you need to have immunosuppressive agents, my dear children, and cyclosporin A is one such immunosuppressive agent, okay, which is used in organ transplant patients. So suppress the immune system, otherwise they will just fight off any foreign thing that comes in their body, but they do not know that the body needs it. Right? So Trichoderma polysporum releases cyclosporin A. This is from a fungi, please remember it. Now, the third is statin. So first is this, second is this, third is statin, my dear children. Now, statin is being produced by another yeast. Another yeast, or basically fungus, that is Monascus purpureus, and the role of statin is to lower down your blood cholesterol level. It lowers down your blood cholesterol level. How, my dear children? So it acts by competitively inhibiting the enzyme responsible for synthesis of cholesterol. So it is competitively going to inhibit what? The enzyme which is responsible for what? Synthesis of cholesterol. It does not allow the formation of cholesterol to happen properly. So that is the role of statins. So I remember statin by stabilizing the cholesterol. That is how I remember it. So it is responsible for lowering the cholesterol, and it is being produced by Monascus purpureus, which is also, as I said, is a yeast. It is what? A yeast. Saccharomyces cerevisiae is also yeast. This is another yeast which is known as Monascus purpureus. So how do I remember all the names? Is that I put in a category. So this is acid, all the things, okay, acetic acid, butyric acid, citric acid, lactic acid, and things like that. Now over here I have all the products: streptokinase, cyclosporin A, and statins. All the three of them have different functions. Streptokinase by bacterium streptococcus, okay, cyclosporin A from Trichoderma polysporum, which is a fungi, and then I have what? Statins, which is by Monascus purpureus, which is also a yeast. Cool? That is how we have to remember them. Now, going ahead with sewage treatment, everything that is given over here from primary treatment until this particular thing is really important. Okay, I'll tell you what is more important. They will ask you about, apart from asking about the how is sewage treatment done, they will definitely give you a question on how is sewage treatment done. So there is primary treatment, then there is secondary treatment, my dear children. Okay, so basically they will ask you what is biochemical oxygen demand, what are flux, and things like that. So you should be knowing that basically in primary treatment you have two things. So you depend on filtration and you depend on sedimentation. Filtration as in the things which can be filtered out, they just float up, no, they can be taken out. That is filtration, you are filtering out. Sedimentation means the larger things can be sedimented down. So whatever you get in between, the fluid thing that you get in between, what do you call it as? You call it as primary effluent. Okay, you call this as primary effluent. And now this primary effluent which does not have any waste, you are going to take it up for the next treatment, that is secondary treatment. And the things that are left behind, okay, they are known as primary sludge. Okay, the solids and everything that sediments down is known as primary sludge. So primary treatment involves two things, filtration and sedimentation, as I have written over here or marked over here. Do not forget, these are two important points over here. Then you take the primary effluent, that is a fluid, and you are going ahead with secondary treatment, where the first thing that you do is you give oxygenic bacteria, the aerobic bacteria, okay, that needs oxygen. So what it does is that it is now going to break down a lot of things. This aerobic bacteria which needs oxygen is going to break down a lot of things, and while doing so it is going to create a flock. What is flock? Flock is basically a thing that is being formed, a mass of bacteria associated with fungal filaments to form mesh-like structures. So these things are there. These bacteria are really important for breaking the organic compounds there. So that is why we say that flocks are there. Basically the bacteria's amount is also increased, okay, since aerobic condition is provided and the food also you are providing. So they will create a mesh-like structure along with the fungal filaments, and this is what you call as flocks, my dear children. So these bacteria will keep on breaking down organic compounds, okay, using oxygen. Now the amount of oxygen needed by the bacteria to break this organic compound down is known as biochemical oxygen demand. Biochemical oxygen demand. Suppose the amount of waste is way too much, that means they are going to need oxygen also to break this down. So can I say the biochemical oxygen demand is too much? Yes, demand is way too much. Okay, now suppose the waste is less, organic compound is less, the amount needed for breaking down this will also be less. So can I say biochemical oxygen demand is less? Yes. So basically the secondary treatment goes on until the biochemical oxygen demand is reduced, my dear children, until it is reduced. If it is way too much, that means there are too many bacteria, that means there is too much waste also to break it down. And when it happens, the bacteria is taking down all the oxygen, but what about the aquatic organisms which are there? It is not, they are going to get harmed because of this. They are not getting it enough amount of oxygen. So this is also a bad problem, okay, very big problem. That is biochemical oxygen demand, if it increases, that means it is a dangerous situation. So aerobic treatment goes on until basically the biochemical oxygen demand or BOD reduces. So you have to be knowing about flocks and also BOD. There might be separate definitions coming for this, so you should be knowing about that. Okay, now once the BOD of sewage or wastewater is reduced significantly, the effluent, again the water content which is there, is then passed down into settling tanks where bacterial flocks are allowed to sediment. So it is going to settling tank, that is the next thing, and there the bacterial flocks and everything which was there, it will settle down. The sediment is called activated sludge. Okay, whatever is sedimented down, you call it activated sludge. Now this has the bacteria and everything, so what do? You take a little portion of this thing and you put it back again into the other one, so that the bacteria can grow again for the next treatment. Okay, so small part of the activated sludge is pumped back into the aeration tank to serve as the inoculum as a starter material, okay, so that they can do the rest of the work. So this was the aeration tank in which everything was done. Now you pass into settling tank, okay, where everything settled down and you have activated sludge as a sediment over there. Now the remaining major part of the sludge is pumped into large tanks called anaerobic sludge digester. So in the settling tank, you made them settle down, rest of the things are still left, you're going to pump it down into what? Anaerobic sludge digester. Anaerobic, okay, there is no oxygen over here. So here when there is no oxygen, my dear children, they are going to again work on the organic component, everything which is still left behind, okay, and these bacteria are going to eat the other bacteria which is aerobic. They are also going to digest on the fungi and everything which is there. Little portion of it you used for preparing as inoculum, rest of the thing is still left, right? So everything is going to be acted upon by the anaerobic bacteria now in the anaerobic sludge digester, my dear children. During this digestion, the bacteria produce a mixture of gases such as methane, hydrogen sulfide and carbon dioxide. You have to remember the names, okay? Methane, hydrogen sulfide and carbon dioxide. 3 gases are being produced by the anaerobic bacteria. That means you're talking about methanogens probably. Methanogens are the one that produces methane gas as well as hydrogen sulfide as well as carbon dioxide. Now these gases, they form the biogas. Okay, so gases which are being produced by the anaerobic bacteria, it forms the biogas which can be used for any other work in kitchen or things like that, and the rest of the things which are being, so the effluent which is going to be produced after this, okay, the watery thing which is going to be produced after the treatment is pumped back into the stream and the normal water bodies and things like that. Okay, so we made biogas out of it, okay, and now we have the normal water also which is being pumped back. Rest of the things which is not needed might be utilized as fertilizers or stuff like that. Cool. So you have to know about all this thing. As I said, flocks and BOD is something that is very important. You have to go through it once more. The definition comes in a separate way. Okay, now microbes in the production of biogas. Now this is very much important. However, certain bacteria which grow anaerobically on cellulosic material produce large amount of methane along with CO2 and H2. So basically talking about methanogens. So that means there are certain bacteria which are going to work anaerobically, the one that I spoke about right now. Okay, they are going to work in a condition where there is no oxygen. Okay, and these are known as methanogens. The common bacteria that we have is methanobacterium. Okay, methanobacterium. Now this methanobacterium, they act on cellulosic material, as in the one that has cellulose. Cellulose is a thing, carbohydrate that we have, my dear children, complex carbohydrate, and it works. The methanogens work on it and they produce methane, and they produce what, carbon dioxide, and they also produce hydrogen gas. That is what is being said. So these bacteria are also present in the rumen of the cattle. It is present in the rumen of the cattle, which is a part of the stomach, my dear children. So it is there. Might be helping in what, breaking down of cellulose. In the rumen, this bacteria help in the breakdown of cellulose and play an important role in the nutrition of the cattle. Okay, so which is why when they put out the dung, okay, that dung also has what, my dear children, methanogens. And now suppose I have the dung, I am going to treat it further for giving me biogas. What is biogas? It is methane and everything together, right? So obviously this dung has it, and now I am putting it in an anaerobic condition where there is no oxygen. I am putting in that particular condition. Obviously the bacteria which are there, they will be growing and they will be producing more and more of what, methane, CO2 and H2, and basically this is what is known as biogas. And this is how the biogas chambers are running in our country. So this is the biogas chamber. Here I am going to put the dung and water. This is going to go in there in the digester, my dear children. You have to draw this diagram. This is really important. Question is asked to draw the diagram of biogas plant. Okay, so this guy is having the dung and everything. I'm going to agitate it a bit, and then things are going to go up over here. This is anaerobic condition. There is less amount of oxygen in there, right? And now during this particular condition, it will be releasing what, methane, it will be releasing carbon dioxide and also H2. And this thing is, as you can see, there is a pipeline over here, a gas pipeline. This will be giving biogas for the usage in the households and stuff like that, for production of electricity and things like that. And now whatever is left behind, it can be taken into a sludge, okay, some next part of it, and this sludge which is there, my dear children, it can be used for production of fertilizers as well as what do you call, manure and things like that. Okay, so biogas plant would be asked, so please make sure you know it. So biogas plant consists of a concrete tank that is 10 to 15 feet deep, in which bio waste are collected and a slurry of dung is fed. A floating cover is placed over the slurry, which keeps on rising as the gas is produced in the tank due to microbial activity. The biogas plant has an outlet which is connected to a pipe to supply the biogas to nearby houses. The spent slurry is removed through another outlet and may be used as a fertilizer, as I said. Now over here, the technology of biogas production was developed in India mainly due to efforts of Indian Agricultural Research Institute and Khadi and Village Industries Commission. This is also a question which is asked, who is responsible for the new technology of biogas? So you should know, it is what, Indian Agricultural Research Institute and also Khadi and Village Industries Commission. Cool, KVIC. Two things. Indian Agricultural Research Institute and KVIC, Khadi and Village Industries Commission. So don't just remember this and go. They might give you something very, Khadi and Village, something else commission or corporation or something like that. So I want you to write it and practice, but this is important. Okay, question will be coming on this as well. Now going ahead, you have biocontrol agents. That is the last part of the chapter as far as I remember. I think, yes, no. Yes, no, okay. Anyways, so biocontrol agents are the ones, basically, for example, we are putting pesticides to kill the pest, right? So instead of putting pesticide, can't we use organisms only which will be feeding on other organisms which are not really needed in our environment? Yes, not exactly, not needed. Basically, we should be these biocontrol agents, these organisms would be feeding on them and making sure that the level of them is greatly reduced in our environment, because they are not good for the plants and the things like that. Okay, so those are known as biocontrol agents. So examples are given, you have to remember all the examples. The very familiar beetle with red and black marking, which is known as ladybird, my dear children, and dragonflies are useful to get rid of aphids and mosquitoes. So dragonflies which are there, it feeds on the larvae of mosquito, whereas when you talk about aphids, aphids are being eaten up by ladybird. So this is gonna come, you have to be sure that you know about this. Ladybird to eat aphids, dragonfly as a biocontrol agent for mosquitoes especially. Okay, now butterfly caterpillars is to control butterfly caterpillars is the bacteria Bacillus thuringiensis, BT. Okay, so for insect caterpillars and things like that, for certain worms you have Bacillus thuringiensis. These are available in sachet as dry spores which are mixed with water and sprayed onto the vulnerable plants such as brassicas and fruit trees, where they are eaten by insect larvae. So my dear children, insect larvae is being usually dealt with the help of what? Bacillus thuringiensis. Okay, now how it is being put? Basically the spores of this is being put in the water and is being sprayed upon. So I'll tell you how it works. So Bacillus thuringiensis, it produces an inactive toxin. What is it? Inactive toxin it is producing in its body. Now if suppose I am an insect larva, I ate this bacteria, what happens is that this inactive toxin is in my body now, but when it reaches my gut, okay, I am an insect larvae, and when it reaches my gut, my dear children, in this gut, this particular inactive toxin will convert into active toxin and it is going to cleave away my epithelial cells and I am going to die. That is how it works. Okay, so the toxin is being released and the larvae gets killed because of this. Scientists have introduced Bacillus thuringiensis genes into plants as well. The genes which is responsible for producing this toxin substances, I can take these genes and been put into plant, and plant itself can produce it. It does not have to wait for the bacteria to help it out, right? So that can also be done. Now, a biological beta cotton is an example of such plant. Okay. Now, a biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma polysporum, remember, it was the one that produces cyclosporin A. Now, the same Trichoderma species, you know what? They are fungi, okay, and they are the ones that are going to kill the free living fungi which are very common in the root ecosystem. So, in the roots of certain plants, no, this particular other fungi, okay, not this fungi, other fungi is going to cause a huge ruckus, and to kill them we have Trichoderma. So, Trichoderma basically focus on the root parasites and basically kill them off. Okay. Now, apart from them, you have baculoviruses and are pathogens that attack insects and other arthropods. So, baculoviruses, again, they are the ones that attack insects and arthropods, so they can also be used. Apart from that, nucleopolyhedrovirus, okay, oh, wait. Okay, so this baculoviruses that you see, they are coming under the genus that is nucleopolyhedroviruses, and these viruses are excellent candidates for species specific narrow spectrum insecticidal application. As in, if you are worried about one particular species, how to kill it, okay, so they are the ones that specifically kills those species only, it does not hamper other species which are running around. So they are narrow spectrum, as in they do not have a broad spectrum, they can kill this, no, it's not like that. They have a proper thing, they will be killing only those organisms which they can, okay, and those are, so baculoviruses are the ones that will be killing insects and arthropods, and they come under the genus nucleopolyhedroviruses, okay. Now, apart from that, what do we have? We have biofertilizers, which is the last part of the chapter. Yes, this is what I was talking about. So, again, we put chemicals, we put phosphorus, we put iron and things like that, okay, which can be synthetic or not synthetic. But instead of that, can't we just rely on organisms which can also fix nitrogen or phosphorus and stuff like that for the plants? So, those are known as biofertilizers, my dear children. So, biofertilizers are organisms that enrich the nutrient quality of the soil, and the main sources of biofertilizers are bacteria, the fungi and the cyanobacteria. Okay, the main group of organisms which come under biofertilizer category are bacteria, fungi and cyanobacteria, which is same thing. So, what do they do? The nodules of the roots of the leguminous plants formed by symbiotic association of rhizobium. So, what does rhizobium do, my dear children? Rhizobium is the one that fixes nitrogen. So, isn't it a good thing? Yes, it is a good thing. So, rhizobium is acting as a biofertilizer for the plant. Now, rhizobium is a bacteria, you have to remember it, and this fixes what? Nitrogen for the plants. Other bacteria that can fix nitrogen, atmospheric nitrogen, while free living in soil is azospirillum and azotobacter. Okay, azospirillum and azotobacter are also something that can fix nitrogen free living in the soil. Now apart from that, you have fungi. Fungi are also known to form symbiotic association with plants, my dear children. What does fungus do and what does plants do? You know, plants perform photosynthesis. In this way, they are producing organic compounds. Now fungi can have these organic compounds, but in return, what does the fungi do? Now fungi has small hyphae which can go deep into the soil and take the nutrients. So this fungi take in the nutrients and give to the plant, whereas plant gives some food to the fungi as well. Now, and this association that they form is known as mycorrhiza. Now you have to know, and this is a question that is being asked, the fungi which does this, they belong to which genus? So they belong to the genus Glomus. Okay, they belong to genus what? Glomus, my dear children. Please remember that. Now apart from that, you have some more thing who can fix nitrogen, that is Anabaena, Nostoc, Oscillatoria, etc. In paddy fields, cyanobacteria serve as an important biofertilizer. Blue-green algae also adds organic matter to the soil and increase its fertility. So all these examples you have to remember it. Again, Azospirillum you remembered, Azotobacter, again Rhizobium also. Over here you have Anabaena, Nostoc, Oscillatoria, everyone, all of them, they are fixing nitrogen, my dear children. So they are all biofertilizers and they are all bacteria. In paddy field, you have cyanobacteria. Please remember that it is going to act like a major biofertilizer. Now blue-green algae also adds organic matter to the soil and increases its fertility. So all the small details which is there is kind of really important, and with this we are completing the chapter that is microbes in human welfare. You tell me, was it difficult? Was it way too difficult? No, I don't think it was way too difficult. Now I'll tell you when it becomes a lot more easy than this, when you practice questions. Okay, the time when you practice questions, it becomes lots and lot more easy. So I'll definitely recommend you to practice the questions from Master series. Go watch that. It will be really beneficial for you to understand this chapter really better. Yeah, so that's all, and do let me know if you have any doubts. I am here to solve and take away all your doubts. And I hope that you are happy, stay blessed. Bye bye, take care. Do not forget to subscribe to our channel, Diksha Karnataka.